Millions of years ago, in a magical land far, far away, otherwise known as Alberta, Canada, there lived an unusual dinosaur by the name of Stegosaurus vilidum. Stegosaurus is part of the Pachycephalosaurs, or Pachyes for short, which were herbivores known for their thick, helmet-like skulls, used as a visual display and for protection during head-butting contests, much like the bighorn sheep of today. This study analyzes airflow patterns through the nasal capsules of modern-day birds, crocodiles, and lizards as guides to determine the possible location of nasal soft tissue structures in extinct taxa, such as Stegosaurus. In order to accomplish this, the researchers used the interdisciplinary engineering-based field of computational fluid mechanics to model how air flowed through some of the dinosaurs' closest modern-day relatives. Once they had analyzed airflow patterns in these species, the researchers turned back to Stegosaurus and modeled its nose with soft tissue schema similar to its extant relatives. This particular dinosaur was selected because of its skull thickness, nearly 2 inches, thick enough to allow for the ossification of soft nasal tissues in the olfactory region. Their thickness also allowed for Stegosaurus skulls to survive the ravages of time much better than the rest of its skeletal structure, which made scans of their skulls particularly useful to scientists. CT scans of Stegosaurus' skull were taken at the University of Texas High-Resolution X-ray CT facility. These scans revealed bony turbinates in the dinosaur's olfactory region, bony ridge in the nasal cavity of the Stegosaurus, which could act as a support for cartilaginous turbinate structures like the ones in extant vertebrates' nasal passages, which, in addition to regulating airflow, prevent water loss in exhalation and cool the blood. In order for the researchers to determine if the clues they found actually indicated the presence of soft tissue turbinate structures in the living Stegosaurus, computational fluid dynamic techniques, or CFD for short, were used to measure and track the airflow through the Stegosaurus' nasal passages.
To build their airflow models for testing, the group consulted previously researched data covering much of the extant phylogenetic branch of dinosaurs. Using these data to make some assumptions about their respiratory variables, they further referenced existing literature to arrive at their viscosity model for airflow, as well as the steadiness of flow. With these parameters in place, they were ready to use their CFD software on their high-resolution skull reconstructions to simulate airflow as it might have been in a living creature. This model of airflow was applied to the CT scans of skulls with and without the presence of turbinate structures. When the researchers analyzed the airflow patterns for the skulls lacking turbinate structures, that is, where soft flesh hugged tightly to the nasal passageway, they found that air moving through the nasal passages completely missed the olfactory region. The olfactory region is the sensory system responsible for an organism's sense of smell. If air was not reaching the region, then the organism would not have possessed a sense of smell. This was unusual since Pachyes should possess a keen sense of smell due to the relatively large olfactory lobes present in the brain, but nevertheless, air was not passing through. This led the researchers to add 3D structures modeled after turbinates into the virtual nasal cavity, as well as different conchi patterns taken from members of the Stegosaurus' extant phylogenetic branches, such as birds, lizards, and crocodiles, to see if this helped solve the airflow issues. And it did. With the addition of turbinate structures, the air reached the olfactory regions and passed over major blood vessels for the purposes of cooling the brain before continuing down the lungs for respiration. Among the various styles they tested, the branched conchi, like those possessed by modern ostriches, sent the most air through the olfactory region upon inspiration. This suggests that the dinosaur Stegosaurus likely possessed turbinate structures and branched conchi within its nasal cavity for the purpose of regulating airflow.
This research is important, especially to our Biology 1520 coursework, because it reaffirms the evolutionary tree for modern-day extant species of birds and crocodiles that possess similar turbinate structures, which touches on our studies of phylogeny and fossil records. It also shows that computational fluid dynamics can be successfully used to direct airflow through a skull to determine the path that air needs to take in order to cool the brain and pass through the olfactory regions for sense of smell. In that respect, the study heavily relies on our knowledge of the circulatory and respiratory systems, and what's more, it taps our knowledge of the sensory processes, such as olfaction and thermoreception. Future research that could be done to further this investigation would be to look at the processes by which the dinosaur brain is actually cooled, and to analyze other dinosaurs with more complex skulls and olfactory structures to see if they too possess turbinate structures for the purposes of bolstering and analyzing the phylogenetic trees of dinosaurs.